Every day, animals are forced to undergo uncomfortable and often painful experiments that yield little to no beneficial scientific results. According to the Animal Welfare Act that was passed in 1962, animal testing is the practice of using non-human subjects for scientific research purposes. For years, household products, pharmaceuticals, and cosmetics have been tested on animals. An estimated 50 to 100 million animals are used worldwide each year for experimentation. These animals range from birds, rats, mice, reptiles, amphibians, fish, and invertebrates. And despite the fact that there are several animals that are not legally permitted for research, such as dogs, cats, human primate mammals like monkeys or the like, and all other warm-blooded animals, they are all victims of human cruelty by research, testing, and experimentation. Under the AWA, animals can be starved, electrically shocked, driven insane, or burned with a blowtorch as long as it's done in a clean laboratory. There is not enough being done to prevent animal cruelty, and though the AWA prohibits this animal brutality in writing, unlawful actions towards animals is still frequent. Because the people that enforce the act are affiliated with animal research agencies, their lack of objectivity makes the act severely under-enforced. Our major goal is to preserve animal rights, which leads me to our main claim that animal testing should be abolished by eliminating all cosmetic and medical research on animals. Our first secondary claim is that animal testing is unethical, and our second secondary claim is that animal testing is unreliable. Animal testing is unethical because most experimentation is abusive. In 2007, Monaim A. Fadali, writer of Animal Experimentation, A Harvest of Shame, and UCLA clinical staff reported that John Drees helped develop a method to assess eye damage and skin irritation from chemical warfare agents. The Drees test continues to be used today for cosmetic products like shampoo, hairspray, deodorant, and detergents, and the test typically involves putting a chemical agent in one eye of a rabbit to measure the amount of deterioration of the eye. In the act of doing so, rabbits are immobilized in full body restraints while the substance is dripped or smeared into their eye or is rubbed onto their shaved skin. This usually results in screams of pain, and many break their necks in attempts to break free. A Tufts University study by Director of Center of Alternatives to Animal Testing, Alec Goldberg, in 1970, proved that the rabbit experimentation grossly overpredicted the effects that could be seen in the human eye, and does not reflect the eye irritation hazard for humans. It also proved that the human 4-hour skin test patch has proven to provide chemical skin irritation data that is far more accurate than the inhumane tests that are done on rabbits. So essentially, these researchers are torturing innocent animals for inaccurate test results. Our second secondary claim is that animal testing is unreliable. Animals are not anatomically similar to humans. Alka Shanana, PhD, laboratory oversight specialist, reported that of all drugs that test safe and effective on animals in laboratories, 92% are found to be either unsafe or completely ineffective in humans, which is because of the fact that they are obviously dissimilar. Thalidomide is one of the several drugs that was tested in animals but were detrimental to humans. In the Gold Coast Bulletin of April 1995, an article reported that in the 1950s, thalidomide was marketed as a wonderful sedative for breastfeeding and pregnant mothers. Despite its safety testing on animals, at least 10,000 children whose mothers had taken thalidomide were born with severe deformities. Cleoquinol is another example of a drug that was safely tested in animals and had a severely negative impact on humans. This drug, manufactured in Japan in the 1970s and 80s, was marketed as providing safe relief from diarrhea. Not only did cleoquinol not work in humans, it actually caused what it was trying to prevent, which ended up being the least of their problems. As a result, cleoquinol became administered to the public. Some 30,000 cases of blindness and or paralysis and thousands of deaths occurred. Clearly, if we are going to make medical progress, a new approach is needed. Human medicine can no longer be based on veterinary medicine. In general, it is dangerous to apply data from one species to another, and there have been several tests that demonstrate how different animals react to things in comparison to humans. Penicillin kills cats and guinea pigs, but has saved many human lives. Arsenic is not poisonous to rats, mice, or sheep, and digitalis raises blood pressure to dangerous levels to dangerous rates in dogs, but continues to save countless cardiac patients. by lowering their heart rates. Even though pharmaceuticals are routinely tested on animals, the Journal of American Medical Association reported that 100,000 people every year are killed by prescription drugs, and more than 2 million are hospitalized as a result of prescription drugs that have been tested on animals. So animals not only react differently than humans to different drugs, vaccines, and experiments, they also react differently from one another. Ignoring this difference has been and continues to be very costly to human health. Animal testing must be discontinued completely because it not only is unethical, but it is also proven unreliable.